Este es el ritmo de Latinoamérica.
This is a review of Mitosis and Meiosis by Hannah and Sully.
Mitosis is the asexual reproduction of cells. The function is it helps an organism grow by making new body cells, also known as autosomes. Autosomes undergo this process. 2 daughter cells are produced. There is no change in chromosome number. There is 1 division in the nucleus, and the DNA between the parent cell and the daughter cell is identical.
Meiosis is sexual reproduction, and it makes gametes, which are sperm and egg cells. The type of cells that undergo this process are gametes. They are located in the testes and ovaries of the body. 4 daughter cells are produced. The number of chromosomes is reduced by 50%. There are 2 divisions in the nucleus. DNA is different in the daughter cells because of crossing over.
Somatic cells in mitosis make up our body tissues and cells such as hair, skin, and lung cells. These 2 somatic cells divide to make 4 new cells. The 4 new cells make 8 new cells. Somatic cells are also eukaryotic, which they contain a nucleus. Somatic human cells have 23 pairs of chromosomes, or 46 chromosomes. This is a diploid cell, which means it has the full number of chromosomes. The 4 stages of mitosis are prophase, metaphase, anaphase, and telophase, and it also goes into cytokinesis.
Meiosis serves 2 important purposes. It keeps the number of chromosomes from doubling after each generation, and it provides the genetic diversity in offspring.
Prophase in mitosis is where chromosomes condense and become visible. The spindle fibers emerge from the centrosomes. The nuclear envelope breaks down and the centrosomes move toward opposite poles. The next step is prometaphase. Chromosomes continue to condense. Kinetochores appear at the centromeres, mitotic spindle microtubules attached to the kinetochores. In metaphase, chromosomes are lined up at the metaphase plate. Each sister chromatid is attached to a spindle fiber originating from opposite poles. In anaphase, centromeres split in 2. Sister chromatids, now called chromosomes, are pulled toward opposite poles. Certain spindle fibers begin to elongate the cell. Next step, telophase. Chromosomes arrive at opposite poles and begin to decondense. The nuclear envelope material surrounds each set of chromosomes. The mitotic spindle breaks down. Spindle fibers continue to push poles apart. Lastly, cytokinesis is where it separates the 2 daughter cells.
Meiosis starts in interphase 1, where cells undergo a round of DNA replication, forming and duplicating chromosomes. Each chromosome pairs with its corresponding homologous chromosome. Spindle fibers attach to the chromosomes. The fibers pull the homologous chromosomes towards the opposite ends of the cell. Nuclear membranes form, and the cell separates into 2 cells. Meiosis 1 results in 2 haploid daughter cells, each with half the number of chromosomes as the original cell. The chromosomes line up in a similar way to the metaphase stage of meiosis. The sister chromatids separate and move towards the opposite ends of the cell. Meiosis 2 results in 4 haploid daughter cells.